Hey guys, this screencast I'm going to be going over the movement of an action potential, saltatory conduction, and the all or none response. And the outcome that we're covering today is going to be explaining the formation and transmission of an action potential. So today's focus will be the transmission part, all or none response, and the intensity of response. So this builds on last screencast where we take a look at, what is an action potential in a localized portion of an axon. Now we're going to take a look at how, a action potential is like a series of action potentials, moving along the axon. So one way that it's often described is as a wave of depolarization. And I like to use the analogy of falling dominoes, as you can see in the bottom right-hand corner here. So essentially, if this first domino is like an action potential in a localized area where you have depolarization, sodium ions rushing in, then what will happen is that will trigger, those sodium ions will actually flow down the axon and then trigger an action potential right adjacent to it. So in other words, once you knock down the first domino, it's going to fall and knock down the second domino, which is going to fall and knock down the third domino, and so forth. And so when you're watching falling dominoes fall, you can sort of see that point at which the dominoes are actively falling. And it almost looks like it's, moving, so to speak, right? So that's essentially what's happening with an action potential or a nerve impulse traveling along the axon. We like to think of it as a wave of depolarization followed by a wave of repolarization. So first action potential generated by sodium ions are rushing into the cell, like I said. Okay, that's causing depolarization. So if we look over at the right-hand side, where it's purple here, that's where we're getting depolarization as sodium ions are rushing in. When we look further down the axon, we can see the membrane is still polarized, so still negative 70 millivolts. And then those sodium ions that rush in are attracted to the negative charge right adjacent to it. So those sodium ions flow over to the next segment of the axon. And that's what's going to trigger, that's the stimulus that's going to trigger an action potential in the next segment. So as time goes by, then we're going to get the next section being depolarized of the axon. In behind where you're getting depolarization, then you're getting repolarization. So there's where you get your wave of depolarization. So if we look at the next sequence in time here, the next segment's being depolarized. Okay, the section behind it is being repolarized. So we end up having a wave of depolarization moving along the axon, followed right behind that with a wave of repolarization. Okay, if you look at this diagram, same sort of thing, but I like it because it's got the color added to it here. So again, step one, sodium ions are rushing in, getting depolarization. If we look further down, that's still polarized. And then as the next sequence here, number 2, now those sodium ions had rushed over to the next segment, causing an action potential in the next segment of the axon. And in behind that in green, you can see potassium ions are diffusing out. So that's repolarization that's happening there. And then the membrane further down the axon is still polarized. Okay, so one question that comes up that I've seen, that you could come across is if you have a segment of axon and it's broken up into 3, and we have positive in the middle and negative on either side, could we tell which segment is depolarized, repolarized, and polarized. And if I just draw it like this, then obviously in the middle it's depolarized. But obviously the two ends are negatively charged on the inside, so it's impossible to tell which one is repolarizing and which one is polarized, unless I add an arrow to show the direction of the nerve impulse. So if the nerve impulse is traveling to the right, just like that diagram on the right is, then we would know that this segment right here must be polarized, and the segment in behind there must then be the portion that is repolarizing. I'm gonna erase some of this, making a mess. All right, so flow of sodium ions from the area of action potential towards the adjacent regions of the resting membrane is what's causing depolarization. And then again, right in behind that segment that's just been depolarized, so when I look down on this portion here that's repolarizing, it's currently in its refractory period. So again, it can't have another action potential until it fully recovers and gets back to resting membrane potential. Okay, this is why when you look at a nerve impulse along an axon or along a neuron, it's unidirectional, so it only flows in one direction from the dendrites, cell body, axon, out through the end branch terminals. Okay, because when we look at this picture, again, the nerve impulse can only travel towards the right here. It can't travel towards the left where the membrane is in its refractory period. Okay, so this is essentially what I just talked about. Okay, again, I talked about this in the last screencast in terms of what a refractory period is. So the refractory period is the time it takes for the neuron to get back to resting membrane potential, which is, if you recall, high sodium ion concentration on the outside, high potassium ion concentration on the inside. And one way to measure that again is to go from maximum depolarization to the time that this neuron gets back to resting membrane potential, which is somewhere around here. This time period down here, that's the refractory period. You could measure it on a graph if you needed to. Okay, that's just an animation you can watch. So in previous screencasts, we looked at neuron anatomy, and in those screencasts, we talked about how neurons can either be myelinated. So in other words, they have the Schwann cells that are wrapping themselves around the axon, and also recalling that the Schwann cell itself has two layers. It's got inner layers called myelin, or the myelin sheath. So you can see all these little inner wrappings. You can see over here. And then the outer layer, most outer layer, was called the neurolimma, right, which repairs damaged axons. And then recall that there are some spaces or gaps where you have exposed axons called the nodes of Ranvier. The nodes of Ranvier. So one factor we didn't talk about when we're going over the anatomy of a neuron is how Schwann cells or myelinated neurons speed up nerve transmission. So we refer to this as saltatory conduction. So again, this is going to essentially what happens is the action potential jumps from node to node. So try to explain how this speeds up nerve transmission or what this looks like. And we can do that by taking a look at the next screen here. So in this diagram, we can see the Schwann cells and the nodes of our NVA. And notice that the sodium and potassium gated channels are only located at the nodes of our NVA. And you do not see these ion channels underneath the Schwann cells. Okay, so again, we talk about the myelin as being like an insulator. And essentially what's going to happen is you get depolarization happening here at this node. So sodium ions rush in, those are the little yellow circles. And once they rush in, then they are going to move through underneath the myelin there. They're going to move through the axon to the next node. All right, and reason being is, well, we can talk about diffusion, but also charge attraction, right? Like down here where it's blue here is still negatively charged. And so these little positive ions are moving to where there's a positive charge. And so they flow through the axon to the next node, and that becomes a stimulus that's going to cause depolarization at the next node. So in other words, it's like the action potential skips all these individual action potentials happening here, and it just flows right through to the next node to cause that next action potential. And by doing that, by jumping from node to node to node, that is going to speed up nerve transmission when you compare that to an unmyelinated neuron. So again, if you watch this animation, I can explain to you, visually, how that works. Better than I can explain it on the diagram, maybe. Okay, again, this little animation compares what it looks like for an unmyelinated neuron on the left-hand side to a myelinated neuron on the right-hand side. Okay, so the idea is that one action potential has caused an X, causes an X, and so this is a slow process of consecutive action potentials, and I guess pushing on a balloon would be the analogy to that. Whereas if you look here, okay, step on the balloon and then it just, you're going to get the pressure happening at the next node here, right? So that's representing an action potential at the next node. Then it flows through, action potential here, flows through here. And so rather than getting all these little action potentials one after the other all the way along, it's jumping and it's much more rapid in that sense. Okay, so that is saltatory conduction. I talked about this idea of threshold potential and the all-or-none response in the previous screencast, but I'm going to go over that again just so we are clear in terms of what that means. So threshold potential or threshold level is defined as the minimal stimulus required to cause an all-or-none response in a neuron. So on the right-hand side, and this is what happens when you go from a Google slide into a PowerPoint. You get this happening. It's a formatting issue. Annoying. But in this little diagram here, you guys recognize a retort stand here. And then hanging off that retort stand is a muscle tissue. And then it's synapsing with a motor neuron. So this is our little motor neuron here. And the idea is that you can stimulate this motor neuron, to send a nerve impulse to the muscle, and then the muscle contracts, and then what's being measured is the force of contraction by this force meter here. So what happened was if you stimulate the motor neuron at 1 millivolt, then you got no contraction, which means that this neuron was not firing a nerve impulse, and you're getting no response by the muscle. However, if you increase the stimuli to 2 millivolts, then this neuron is activated. It sends a nerve impulse, and then the muscle contracts at 3 newtons. Then, as you increase the strength of the stimuli, notice that the force of contraction remains the same. So this lends itself to here, the minimal threshold, right? For this motor neuron here to fire, was it over 1 millivolts or around 2 millivolts, right? And then even though you keep increasing the strength of the stimulus, the response ends up being exactly the same. So that lends itself to the idea of the all or none response. So in other words, a neuron will fire maximally or it will not fire at all, as long as you meet the minimal threshold potential. So threshold levels are different for each neuron. So what this little example shows is that increasing the intensity of the stimulus above a critical threshold value will not produce an increased response. Okay, so it's either neurons fire maximally or they do not fire at all. So we just call this the all or none response. So then the question becomes, how does your nervous system then detect various levels of stimuli? Okay, so for example, your hand, if you dip your hand in ice cold water and then warm water and then hot water, your brain can process those various temperatures. Okay, so how would your brain interpret those various temperatures then if neurons are either just firing or they're not firing at all? And the answer to that is your brain interprets two things, okay, to allow it to differentiate various levels of stimuli. So one is the number of neurons that get excited. So for example, if you look down here, we got a glass rod touching a sensory receptor or whatever this is at 40 degrees. Well, maybe one neuron is being excited here and one neural pathway is sending signals to the brain for processing. Whereas if you have a glass rod at 50 degrees, now you have two neural pathways sending signals. And so as you increase the temperature, more neurons are getting excited and sending nerve impulses to the brain. So your brain can interpret that, the number of different neural pathways sending signals to the brain. And then secondly, within each neural pathway, the frequency at which impulses are being sent to the brain can also be processed.